For the question number 1, we are required to identify the genes involved in the production of taxol in Taxus brevifolia using cDNA microarray hybridization, experiment and step involved using a flowchart, and also the confirmation of the gene. For the step number 1, we are required to collect the tissue, in which we collect from the taxol-treated HeLa cell and non-treated HeLa cell. HeLa cell is a breast cancer cell. Isolation of RNA is when we add organic solvent into the sample and then we centrifuge it to separate the RNA in both sample. For the isolation of mRNA, we wash over the sample in column with the oligodetibates that somehow will bind to the poly A tail of the RNA string, and then buffer is added to detach the mRNA. Labeling of the RNA will be using the fluorescent dye of CY5-DUTP into the taxol-treated HeLa cell and CY3-DUTP into the non-treated HeLa cell. The reverse transcriptase will assemble the label nucleotide and then the mRNA will be degraded. For the applied DNA, microarray containing a single-stranded DNA of different genes, in which both samples of cDNA are added into the microarray to hybridize as the gene is expressed. Few strands are not hybridized and will be washed away. We scan the DNA using the DNA microarray analyzer, and then the data will be analyzed, which is the red color superimpose for CY3 and the green color represent the CY5.
Hello! I will talk about the result in microarray. What do you expect the result for microarray? Of course, you want to see the color. Yes, there will be red, green, yellow, and black. But black means nothing because it's only an unhybridized gene, so let's move on. The color that I pick is green. But why? Because that cell gene will attach to HeLa cancer so that it can repress the cancer cells. This is the what we want to see. Red will be appear when taxol gene is hybridized with non-treated cell because there is nothing to repress. How about yellow? No, there's no appear yellow. And here is the candidate's gene along with the accession number to produce taxol for community. Thank you.
Real-time PCR output shows an amplification plot of the gene expression of each sample, but normal PCR only shows the endpoint result of band, position, and intensity. Although real-time PCR saves time and do not require post-PCR processing, it shows high deviation in the same experiment. Thus, a reference gene is needed to normalize the result. After preparing all the necessary materials, it is time to mix them together. Each sample is in their own Eppendorf tube. Pipette the master mix from earlier and also the set of respective primers into each Eppendorf tube. Don't forget to add the props too. Mix them together using pulse vortexing. Next, load the mixture into the PCR plate, normally containing 96 volts. The plate is placed in a thermocycle to amplify the samples. The fluorescent signals produced by probes grow with the amplicons, meaning more amplicons, more signals. The signals then pass through the emission filter and is detected by charge coupled device CCD camera and converted to digital data. Digital data collected is displayed in the amplification plot together with the cycle threshold of each sample. From there, we can determine the gene expression of each sample. The end.
For the experiment steps involved, first, take up cells from Taxus brevifolia culture, then extract and isolate the RNA. RNA is then purified and assessed. After that, a mix will be prepared containing a primer, reverse transcriptase, DNA polymerase, RNA template, which is prepared earlier, real-time PCR buffer, dNTPs, and water. Next, RNA is transcribed into cDNA by reverse transcriptase enzyme, where the cDNA is then used as template for real-time PCR after the probe is chosen, which is TaqMan probe. TaqMan probe is chosen because of its specific hybridization, unlike DNA binding dye. Next, post-PCR processing is eliminated, which reduces assay labor and material cost when using TaqMan probe. And lastly, it is ideal if the results are required in a short time.
Okay, next, I will continue with the question number 3, primer design. So the highlighted gene accession number are the genes that we chose for this question based on genes that are presented before on previous questions. These are the tools that we use on primer designing, which is Primer Blast. Primer Blast is the combination of Primer tree and Blast. So the way this tool works is we blast the desired genes into the Primer Blast, and it will produce a number of possible primer for a forward and reverse primer that we can choose from. Let's look on our first gene, Taxus baccata, Taxus gene for taxidiene synthase, that have 3,890 base pairs. So these are the best results that come from the primer tree blast. The details is TM forward 59.83, TM reverse 60.11, the GC content is 55%, and the product length is 743. Next one is Taxus chinesis taxidiene 5-alpha hydroxylase mRNA. The TM forward is 59.67, the TM reverse is 59.47, the GC content is 50%, and the product length is 453. And the last one is Taxus cuspidata 2-alpha hydroxytane 2-O-benzyl transferase gene. So the TM forward is 60.03, the TM reverse is 59.90, and the GC content is 55%, and the product line is 954. So that's all from us. Thank you.